A pseudotime orders the cells along the river, but it doesn't tell you which direction they're heading. Are they differentiating forward or differentiating backward? RNA velocity answers this. It measures the current when a gene is being actively transcribed. Unspliced mRNA, pre-mRNA with introns still attached, accumulates before being processed into mature spliced mRNA. If a cell has more unspliced mRNA than expected from its spliced level, the gene is being upregulated. Transcription is increasing. If it has less unspliced than expected, the gene is being downregulated. Transcription has slowed and existing mRNA is degrading. RNA velocity uses the ratio of unspliced to spliced counts to infer the direction of gene expression change. Mano and colleagues introduced this concept in 2018. CellRank implements two velocity models. The stochastic model assumes each gene is at steady state. The ratio of unspliced to spliced should be constant. Deviations from steady state indicate velocity. Fast to compute but inaccurate when genes are actively changing. The dynamical model fits a full kinetic model: transcription rate, splicing rate, degradation rate, all time varying. It solves the ode for each gene to estimate velocity. Slower, but much more accurate. Your spatial transcriptomics pipeline uses the dynamical model because glaucoma disease progression involves time-varying transcriptional programs. Project the velocity vectors onto the UMAP embedding. Each cell gets an arrow pointing in its predicted direction of transcriptional change. Visualize as a streamline plot. Arrows averaged into smooth flow lines. Streams converging at a point indicate a terminal cell fate. Cells are flowing toward that identity. Streams diverging from a point indicate a branching decision. Cells are choosing between fates. Themes flowing in circles might indicate oscillatory behavior like the cell cycle. The velocity field on UMAP is the most intuitive visualization of dynamic processes in single-cell data. CellRank integrates RNA velocity with cell-cell transition probabilities. The velocity kernel converts velocity arrows into a transition matrix, the probability of each cell transitioning to each neighbor given its velocity direction. The connectivity kernel uses expression similarity. Your pipeline adds the spatial kernel. Cells nearby in tissue space have higher transition probabilities. CellRank combines these kernels into a single Markov chain, connecting to the board game video. Terminal states are identified as absorbing states. For each cell, CellRank computes the absorption probability, the likelihood of reaching each terminal fate. Color each cell by its fate probability. A progenitor in the optic nerve head might have 60% probability of becoming a reactive astrocyte, 30% of becoming a quiescent astrocyte, and 10% of remaining a progenitor. The commitment point is where the dominant probability crosses 50%. The cell is now more likely to reach one fit than any other. Overlaying fate probabilities on the spatial coordinates of your slide tag data reveals where in the tissue cells commit to specific fates. A spatial map of cell decisions in glaucoma progression. The river flows from source to sea. Pseudotime orders the journey without a clock. RNA velocity measures the current from unspliced and spliced mRNA. CellVelo fits the kinetic model. CellRank converts velocity into fate probabilities using Markov absorption. Your cells are on a journey through differentiation, disease, or response to therapy. These methods map the river,